Our lab is trying to identify new approaches to connect the biophysics of ion channels to patient response to antiarrhythmic therapies. Unfortunately, there's not a lot of drugs that are available to treat arrhythmias, and the ones that do don't tend to work on most patients. So what we're trying to do is figure out which patients are going to respond to which drugs so physicians can prescribe that before patients have arrhythmia, so they can go home and spend less time in the hospital. We use advanced imaging to watch how proteins move and how drug molecules interact with those proteins. We then use tissue engineering models to test different approaches in tissue models. And finally, we do clinical studies where we use advanced data models to connect the parameters that we measure with our biophysics methods and our tissue culture methods to figure out how those are connected to the patient response. I'm usually very happy when my students are successful and when they reach their full potential. When students come to the lab, we iterate through different projects. We try and figure out which project is going to best match their skill set and what they're interested in. And I really like working through that process, getting to know the students, getting to know what they're interested in, and getting to know what they're going to be able to spend a lot of time working on, get excited about. I think that process of getting to know them, thinking about science, getting them excited about arrhythmia research and science in general is something that I've really enjoyed doing. That getting a PhD is more about the process and the papers that you put together, the contribution that you make to science, more than it is about getting 3 letters after your name. The goal isn't to get a PhD, the goal is to make a difference and to learn how to do science, how to be a professional scientist and grow from there. A lot of study of antiarrhythmic drugs is done by biologists and pharmacists, but there's really a lot to be learned from a quantitative approach. So using an engineering background to think about how can we best resolve the interactions that antiarrhythmic drugs have with the proteins that they're working on. How do we connect across scales? So drugs interact at the nanoscale, arrhythmias happen at the centimeter scale. So how do we relate what's happening at the nanoscale to the centimeter scale? And finally, taking the findings that you have at the bench scale and in experiments where you're looking at how proteins move and understanding how those affect patients takes a pretty sophisticated model, and connecting those features to patient response is something that not just anybody can do. And so the preparation that people have as biomedical engineers makes them uniquely suited to study that problem.